Hi again, everyone. I'm sorry I had some disruption. Got disconnected from the machine and the whole recording stopped for the first part. Anyway, we continue here. So we were discussing whole genome sequencing, um, where we're looking at the entirety of the organism's DNA. Now, whole genome sequencing, when we use whole genome sequencing, we are able to construct, again, the genome. And whole genome sequencing, when looking at this whole genome, especially when we may have information about characterized libraries, databases, or reference genome, or all of that information, when you try to put everything together, we can gain insights, important insights about protein coding mutations and regions which are linked to gene DNA sequences. So any alterations or mutations that can change the amino acid sequence of the protein, potentially it could alter its function. So whole genome sequencing can release that. Non-coding mutations and the other non-coding regions can be very important, because in these regions there are important regulatory elements like possibly promoters, enhancers, or other non-coding RNAs. So such mutations can again disrupt these regulatory elements eventually, which may impact gene expression. So this type of analysis for specific, patient specific sample or target can release lots of insights. Structural variants, something we discussed previously, we are referring here to changes that that can happen possibly because of some rearrangements of DNA. This could include again deletions, insertions, inversions, duplications, and so on and so forth. Somatic copy number alterations, it refers to changes in the number of copies of a particular region of a DNA. So within the genome. These are especially important in cancer genomic. Sometimes a region of a chromosome might get duplicated or amplified multiple times in a tumor cell. Tumor generally means, higher growth, some amplification, so on and so forth. Translocations are a type of also structural variants, where a segment of one chromosome becomes attached to another chromosome, and so on and so forth, So sometimes also pathogen detection here can be achieved with whole genome sequencing. So if we are sequencing from a sample, for example, patient's blood or tissue, you might see DNA from the host organism, that is the person, and also the pathogen, that is another organism entering the space. Could be bacteria, viruses, fungi, who knows. So when this whole data is analyzed, maybe we can identify these pathogens, especially when we're trying to look into the whole genome and trying to diagnose some infections. So whole genome sequencing, especially when using next generation sequencing, is comprehensive in nature. We can capture a wide range of genomic alterations. So lots of applications, lots of aims can be achieved with that. In terms of whole genome sequencing workflow, and again, we are repeating these steps across the module, but with slight different flavors to emphasize the importance or, the information that we are, some of the informations that are hopefully helpful for you, Library preparation, there are certainly some steps starting with the genomic DNA, depending on the type of sequencing as well, sequencing technology used. Next steps will be determined. Of course, there's DNA fragmentation, so on and so forth. Then we go to the, so library preparation, again, dependent technology, and then we go to the sequencing stage. We might end up doing cloning. We might end up, having cluster formation, maybe sequenced by synthesis, and so on and so forth, depending on the technology. And then we come to the stage of data pre-processing. We have the FASTQ files. That's now a target for us. We know that's something we handled before in this course, something to expect to see commonly in bioinformatics. And then we want to call something for variant calling to see what are the differences that we may realize given this specific genome compared to what we have as documented information. And eventually we can also have other applications. Again, more about the structural variants. Possibly we end up doing de novo assembly if we don't have a reference genome. So in terms of looking at the alignment, okay, when it comes for analyzing data from next generation sequencing platforms, it's useful to see and introduce you to some tools. Okay, so starting with the BAM file. Okay, and the BAM file is a common format for storing sequence data alignments. It is this binary type of data. It is kind of compact. It helps in saving the space because SAM files that are usually produced are large in space. This means eventually we are, whenever the space is smaller on the disk, there is what's called IOs. So I'm trying to read from the disk. This is one of the more very time-consuming tasks in computer systems. So reducing the size of the file means faster access and read and so on and so forth. So let's go over some of the tools, like skim through them that are here in the slides. We have what's called First we go with BioBAM, which is BioBAM can work with BAM files. Okay, it's a collection of tools. So it's something you can download, install as software, coming and offering lots of functionalities. Can help for sorting, merging, and marking duplicates in the BAM files, among other several other tasks. So we are doing some conversion or processing using this tool. Next, we have the FASTQ, okay, file. Then we can go and do some quality check using FASTQ-C, generate some reports, and so on and so forth. Now we have this step which is using Boros-Wheeler Aligner, or BWA, and it's another software package or tool that we can download into our servers for mapping DNA sequence reads to a reference genome. So remember, here we are trying to do this this process of aligning to a reference genome. I'm very sorry. Okay, sorry. Okay. Anyway, so BWA, there are some software. So when you see here MEM ALN, it means it's kind of the algorithms that are there. Okay, for BWA-LN is an algorithm within the BWA software or tools for Illumina sequence reads up to 100 base pairs. MEM is the newer version for longer reads. and it's usually producing higher quality. Another set of tools, Picard tools. This is again a set of tools that can remove duplicate reads, adding reading groups, and generating some various metrics related to sequencing data quality. So Picard tools can be used in different parts, as you see here in this workflow. One of the very important software tools, which is coming from Broad Institute, is GATK. GATK. It's a toolkit for variant discovery. So maybe some of you asked or had this question: how do we do variant, calling or discovery? Well, you're going to use a software which provides this kind of software, like GATK, gives best practice workflows for calling single nucleotide polymorphism, SNPs, insertions, deletions, and it's widely used in industry as a standard for variant calling. So these are the set of tools that, when handling DNA-seq alignment and kind of downstream and processing under this umbrella of whole genome sequencing, that we can look into. When we start diving into or looking into the genome assembly, reference is certainly a keyword that we look into. Reference is, again, there was some initiative which is going to get us to the question: where do we get references from, or where they come from? So it depends on the interest. So sometimes, there is a consortium, a group of scientists or labs or research centers that come together and share interest to study one of the organisms, and they have investments that or funds to do it, and so they end up working on sequencing and doing assembly, curation as well, with our annotation, which is very important, and then they share these results. So it could be that one research lab was able to release information on some of the species that we can use, or there being a consortium. So reference sequence, reference genomes are indeed, or reference sequence are important. Can make the task easier as compared to what we call de novo assembly, which is more sophisticated. So the fact, again, through this whole lecture so far, if we have access to a reference genome, just use it, okay? So in whole genome sequencing, we're looking at different ways. Either it's by the day we are dealing with different fragments, we're trying to align them together to solve the puzzle. We are dealing with maybe millions or billions of reads to end up with what? A linear genome, or maybe circular, depending on the type of species. Either we're following the clone by clone direction, or the shotgun direction, okay? And so it's about, working with these fragments, trying to put them together. What relates to this, to the jigsaw puzzle, and in fact, the jigsaw puzzle itself can be challenging sometimes. Why? And that's the analogy we want to use here, and also indicate why genome assembly can be sometimes hard, okay? So as you would see here, there are different highlights of different ideas. So we may end up with multiple copies. So while I'm trying to align a specific sequence, and I'm trying to reconstruct that region. And again, we have very short reads. We're trying to grow those and put what's called context, right? Longer DNA sequences. Imagine that we have that area, okay, of that specific sequence, which is repeated across many places of the genome, the DNA. Now, when you construct one, which one we are constructing? This may raise a challenge, actually. When we have multiple copies originally coming from the DNA sequence. We don't know if we're dealing with one or multiple copies or how many. So this is an extra added layer of complexity. We may end up having some missing pieces, some full repetitive regions, like exactly the same region is being repeated several times. And what about the corners? We are not dealing sometimes with circular genomes, so we are not able to, know if this is really the end of the part here and this is the beginning and so on and so forth. Sometimes we have dirty pieces, some mutations or something introduced as part of the processing and so on and so forth, or frayed pieces. So longer reads always is gonna, in general, help us in figuring out the proper construction, and of course more accurate reads, meaning fewer dirty ones, are going to be very helpful, as well as simpler genomes. The genome is very sophisticated. Something we know about that species can lead to different complications and challenges. So let's look into this framework here, where we're starting with Illumina FASTQ read files. Of course, there are a set of tools that we can look into here. What I want to highlight is, especially for de novo assembly, when we don't have a reference, so we can look into these sort of tools in this case. Okay, so what if we want to end up prof that we need to do de novo assembly, or we want to assemble in our lab and we don't have a reference, genome? What can we do? Always remember, regardless if you know the tool or not, we have to look into the quality of the reads. Okay, something could have went wrong with the whatever library preparation, the whole experiment, extraction, and so on and so forth. So this is the first step. There is no question before we go any farther. So we can use tools like Trimmomatic. It is a tool for trimming and quality filtering, for example, Illumina sequence data. We need to know if we're going to move to assembly again, and cannot emphasize this more, that it is very important and essential to clean the data. For example, to remove adapters, low quality bases, and other unwanted sequences. We have to be very careful in everything whenever we discuss, any framework, okay, any analysis, quality check here for alignment. If we have the reference genome, as mentioned earlier, if we don't have the reference genome, and again, check that and do some filtering. Next, we have SPADES, or short for St. Petersburg Genome Assembler. It is for genome assembly, and it's an assembler designed for working with Illumina data. It can handle small genomes and produce high quality assemblies. There are also more updates in recent versions that have capabilities to perform metagenomic assemblies. And metagenomic, these are the samples where we have multiple copies of DNAs, maybe from different organisms or samples or species. Therefore, it would make sense sometimes to look into tools like Kraken for taxonomic classifications. And this tool is going to assign taxonomic labels to short DNA sequences with high speed and accuracy. Later, we will have tools like for quality, again, quality control, like quality assessment tool, or QAST, quality assessment tool. It helps in evaluate the quality of the assembly. So once the assembly is produced, and coming from SPADES, for example. I'm going to also refer you to some other tools here, but once the assembly is out, we need to look into some statistic, okay? We're going to see some of these measures in one of the coming slides. SAM tools is a suite of programs for also interacting with the sequence data and SAM BAM files, looking into sorting, indexing, and extracting alignments, variance calling, and so on and so forth. BowTie is for read alignment, as well as fast and memory efficient tool for aligning sequence reads to long references, and so on and so forth. So while SPADES is assembling genomes from reads, BowTie align reads to a known reference. They are these types of tools. So maybe then we have this reference established from here, and then we can do some alignment and bring some discovery to what we're going to look into. And then you have some other, depending on the specific protocol of the study or the aim, we can have we can achieve different outcomes with specific set of tools. What I want to comment on here in the context of this whole genome assembly is that SPADES is primarily, as I said, is used for de novo assembly. And this is something we have to know through this lecture, that this means that we have no reference genome, okay? So SPADES can assemble genomes without the need for a reference genome, especially for, again, small genomes like bacterial assemblies and so on and so forth. But there are many other tools, and the best choice depends often, always we refer to this, there are different tools, we have to understand them then. Okay, so specific data type like short reads or long reads and the organism of interest may guide us to which tool we can use. For example, there is another tool is called Velvet. Okay, is a very other popular older tool for de novo assembly of short sequence read data. And there are many other tools. For example, there is SOAP. This is another tool, SOAP de novo. SOAP de novo 2, and it's designed for the de novo assembly of large genomes from short read sequences. It has also interesting modules for correction and scaffold construction. And many other tools. Okay, there's CANU. Okay. CANU, C-A-N-U. It's designed primarily for long read sequencing. That's, if you remember, we talked about third generation sequencing, PAC Biosciences or Oxford Nanopore, and so on and so forth, as well as many other tools that are out there. It always, gonna bring us the question, why or which tool we are supposed to use, right? And like there are many options, many parameters, right? Sooner or later you will know that this is where many of the bioinformaticians are spending time on in these kinds of tasks, where they are trying to explore the best setup, the best workflow, and so on and so forth. I may share also some, in one of the coming slides, some hints, but in general, you may end up running a few of those. But again, also keeping up with the literature, some of the benchmark studies that are ongoing to find out the best setup for the experiments that you're working on. So, and then just quickly, again, there are AMR characterization, virulence, gene detection, for example, it's a process for identifying genes in a pathogenic organism's genome to know, the relationship of that, for example, virus, right? Virulence gene encode, if it's causing the disease or what. So some of the tools that can be used there is BLAST. I guess we commented on this before in one of the modules, but again, BLAST is very common and well known, and you can access it through NCBI. BLAST stands for Basic Local Alignment Search Tool. So it's widely used for comparing an input sequence against a database of sequences. That is how we can take that part or that sequence, try to see, is that going to what kind of species? So when you have a database or library of species, you can eventually know, oh, there's a match with high similarity and coverage with some specific virus that suggests that virus genes are there in the organism. KML alignment or KMA is designed to map row reads or contigs against, again, also database. SRS2, it's short read sequence typing for bacterial pathogens, and so on and so forth. So in summary, for example, virulence gene detection is crucial for understanding the potential pathogenicity and looking and understanding the infection mechanism of the microorganisms. So these tools offer different methodologies to, again, or options to detect these genes in genomic data sets. Assembly, keep reminding you, is having a large DNA, doing the fragmentations, going with the sequencing, then start doing the assembly, looking for the overlapping regions. This is the whole thing. And of course, so far, we did not dive into specific algorithms in terms of de novo assembly. Now, alignment, we are trying to align the reads, something we discussed before, right? Different data structures into how we can align things. We have a reference genome. So basically, we are trying to map that with some differences. I would say it is still a bit complicated, but it is probably much easier in terms of a computational solution than compared to the de novo assembly. De novo assembly usually ends up building graphs of these reads and trying to find alignments of these reads. So it's a bit demanding process. When I say graphs, think about social networks and how we can relate some of the nodes and edges to the construction of this interesting data structure to tell us, okay, this read has common part with that read, and so on and so forth. And then we use that kind of map to extract that information, which is, again, is going to look into some path that goes through all nodes or different ideas there. So interesting ideas that were well developed in computer science. Who knows if this is going to change with quantum computing and how, but probably this is one of the areas to contribute to de novo genome assembly. So this could be a very interesting topic to learn more about or think about. How can we find application of quantum computing in de novo genome assembly? There are a few terms that we have to have and to know when we are looking into assembly. Specifically, reads is something we know. Contigs are this assembled part, okay, where we end up with, it's not the read itself, but now we have a stretch of these assembled sequences. So, of course, we are trying to assemble the reads, align them on top of each other, trying to find the consensus of that. Anyway, through these steps, we end up with the contig. Yet, some important concepts are as well scaffolding or the scaffold. Scaffolding itself refers to the process of ordering and orienting the contigs. So the input for scaffolding is the contigs that were derived from the previous step. It's trying here to order and orient the contigs based on different pieces of evidences. there can be gaps in this process. So ends here, these ends are referring to gaps. In other words, we don't know which nucleotide. It is unknown nucleotide. We use N for that, something also referred to in the previous module. Now, part of this scaffolding, which is gonna, as it's trying to order the sets of conjugates, put gaps because couldn't find out what reads that would match these regions. Okay, so we're trying to glue the context and so end up with scaffolds. Eventually we want to go back to the full genome. This is more of a hierarchical strategy. We're aligning things. It makes full sense to go one step at a time, from puzzle pieces to partial puzzle to solving the full puzzle. But gap filling here, which is trying to replace these ends, is one of the steps that can be part of the whole process. Okay, gap filling replaces the ends with actual nucleotide sequences. The question is that how is that being done? Okay, so after scaffolding, these gaps between the contexts, which might arise due to repetitive sequences. Why do we have these gaps? Maybe we had repetitive sequences. We couldn't, again, know that what are these repeated regions, ended up missing some information. Sequencing error, maybe we're not having enough coverage or reads for this specific area, or other challenges that we had through the assembly process. Okay, there are usually various tools to deal with this part. Okay, and towards again, as part of the scaffolding process, we're trying to fill these gaps. There are some sources of evidences that one can go through. So, for example, the set, each of these sources of evidences here that you see in the slides provides different length scales of information. Okay, for instance, mate pair libraries might give information in the order of kilobases. Okay, while PAC and end sequences, 10x genomics, linked reads, and high-C data can give information. Spanning much larger genomic sequences or distances. PACs, for example, is a type, as we know, of cloning and can carry larger DNA inserts, 100 to 300 kilobyte in size. Okay, kilobases, 100 to 300 kilobases in size. So what's happening in PAC? The sequencing the ends of these PACs, both ends provide long-range information for scaffolding. In addition, full PACs can sometimes be sequenced to fill specific or resolve complex genomic regions. So part of the protocol, part of the technology itself, we can enhance the level of information needed to aim for higher, or better gap filling. 10x genomic linked reads, for example, as another source of evidence, it is a technology which involves barcoding high molecular weight DNA and then fragmenting it to produce a library. So fragmentation, and then we end up with a library, steps we went through before. Now reads originating from the same long DNA molecule will carry the same barcode. This is the idea. So this allows for the reconstruction of the original molecule sequence and provides long-range information from short read sequence. So we are able to reconstruct the original molecule sequence as we have tagged this information, we have traced this information, knowing that this is coming from the same long DNA molecule and so on and so forth. Different steps and evidences can be there towards, again, building these scaffolds. Now, there's no question, although assembly is by now we know that it's having lots of applications. There are different ways to do it. Um, and we can, there are lots of tools to build a proper workflow, and all depends and start with these questions of your own need and the project needs. Okay, so now hopefully so far some of the things you can summarize or understand. Depending on the species, do I have a reference genome, then I follow one path. I don't have a reference genome, then I follow another path. Along the way, I understand that to construct the whole genome, there are some technical challenges, of course. So we need to assemble the full genome. We know that this is going to follow hierarchy of context, scaffolds. And so there might be some issues along the way. Depending on the technology, also, that's going to mean different things. Okay, maybe we're dealing with long reads. For example, PacBio on nanopore, you end up with more than 10,000 base pairs reads, very long reads. However, they usually come with high error, 5 to 15%. So the computational challenge is how to avoid, how to overcome the error rate with these long reads and so on and so forth. Short reads coming from technologies like Illumina are high accuracy, very high throughput. But again, because they are short reads, it limits the ability to resolve the repeats. We are talking about short overlaps with these short reads. Sometimes it's between 50 to 60 base pairs. Therefore, we may have false matches and so on and so forth. Now, again, there's no question that assembling the genome is a complex task. We may end up with some hurdles or challenges. For example, repeats or low complexity genomic regions, repeated sequences, which as I mentioned earlier, especially those longer than the read length. Okay, that's what is meant by these repeated sequences. If they are, longer than the reads that we are receiving, because of the sequencing technology, are very difficult. Because the same sequence appear multiple times in the genome, we cannot determine easily where in the genome each instance of the repeat belong to. We may end up saying, okay, they are the same thing. Okay, how do we know that we have multiple repeats? And that is actually starting with this question. How do you know that we have multiple repeats? That is a challenge. And once we have multiple repeats, where are they in the genome? What are the locations? What about heterozygosity? Looking at alleles, versions of the gene. So if we have a variant of that gene in diploid organisms, for example, one chromosome could have a variant, example, an A at a particular position, and the other chromosome has a different variant. For example, instead of A, it's going to be G. So this also adds complexity and some hurdles for doing the genome assembly. Sequencing errors. Sequencing technologies are not perfect. We know this. We can end up with substitutions, insertions, deletions, and so on and so forth. Some sequencing technologies actually have difficulties with certain base compositions, especially with regions that are extremely GC rich or GC poor. For example, in PCR amplification, GC rich regions might be preferentially amplified compared to AT, adenine, thymine rich regions, and so on and so forth. So anyway, addressing these challenges often requires a combination of multiple sequencing technologies, specialized assembly algorithms, and rigorous validation and quality control. So that's why I mentioned, even if the genome, again, until we have the ultimate technology, what's going to produce for us, everything that we need, so far it is demanding process in terms of human, resources needed for that, in terms of, discussions, making sure every step is well done. That's why usually, we end up having consortiums or centers working on assembly. But again, doesn't mean you cannot do it. Of course, you can follow the steps. And one person, and we had this in our lab, we had the species, we had the samples coming, and we did de novo assembly. You have the pipeline, and you go through it. That's the pipeline documented in the literature. These are the updated versions of these tools. These are the recommended parameters. Okay, start with it. Run it for a few hours, couple of days on some server, done. We have but again, it's going to be also how much we gain by the end of the day, and notations, quality, and so on and so forth. So it's iterative process. We need to keep enhancing our findings until, we really achieve the outcomes of the studies. This refer here to some biases giving the different technologies and so on and so forth. So now, in terms of a reference-guided assembly, we're trying to align the reads until we get a layout of a group of aligned reads. Then we can derive the consensus. Eventually, the consensus, which is, in the consensus, we are not looking at the reference sequence. We're looking at the layout of these aligned reads. So consensus means that let's take the majority kind of vote whenever we have the option. So here we don't have options, so we can just go with T. Here we have some T's and one A, so majority is T, so we go with this. Majority is C, so we go with this. And then eventually we are looking at the consensus here. So then we can compare these mutations, try to do analysis. That's when we look into what those mean, what those changes would mean. That's kind of the variant calling process. And we have been discussing, of course, de novo assembly, which refers to the process of reconstructing an organism's genome from the sequencing data without using a reference genome. So some of the things that we see here is I want to refer to quickly is assembly with parameter set. Okay, choose appropriate parameter set. Assembly algorithms use the overlap between reads, right, to stitch them, to connect them, to match them together. So if the parameters are not set appropriately, sequencing errors or biological variations can disrupt this process, because we know that there has been some issues or probably some sources of some of these issues. So how do we deal with this parameter? And this is here where I'm going to give you some suggestions, okay, general suggestions. How do we find the right parameter set? And what are parameters? So let's have this as a treasure hunt question. When I say parameters, what comes to your mind? Okay, please write it down, share it with me. So parameters are, of course, options for all of our software pipelines. For every software pipeline, you will find some set of arguments that you need to pass. So if we want to know what we should choose, right? Usually you can start with the recommendations. Assembly tools usually come with default settings, so you can start with those as often. What I would say, it's a good start, okay? Especially if you are unfamiliar with the software, start with it. And then what I've referred to earlier is test and reiterate, okay? Iterate. This is a continuous process of trying to work with different parameter sets and then compare the output. Let's look at some accuracy measures like N50. I'm gonna explain this in one of the coming slides, if not the next one. So contact count, okay, and so on and so forth. So we have some measures to try to see the quality of the different different parameters. Sometimes we know something about our own technology, about the depth of sequencing we have, the complexity of the genome. All of those are source of valuable information that we can can get our attention and want to focus on in terms of the data, your data. Okay, what do you know about your data? Okay, also, what about even doing leverage of multiple assemblies? Multiple assemblies. Okay, so maybe not just running different parameters and that's it. Maybe to run multiple assemblies with different parameters and then use meta assemblers, some kind of assembly reconciliation tools to integrate all these results. Of course, always stay updated in this field. As I told you, it is still an active area of research. Still lots of challenges with it. Maybe you have new algorithms, so keep reading from the top venues. What are the new tools and advances? Always look into talking to the experts, okay? And so here, seek the expertise. You are going to become an expert. You are an expert. But again, nothing wrong to for critical projects to have some consultation and talk to other friends, especially in this interdisciplinary field that we are working with, the domain of bioinformatics. So in de novo assembly, in a nutshell, we are trying to work only with reads. We don't have a reference genome. Eventually, we're trying to find match between the ends of the reads. So maybe this is one read, this is another read. So I'm able to match this part here, match this part, so I can end up with some information in the middle. So kind of, start putting this, grouping these together, deriving longer sequences, and so on and so forth. Now, there are measures to indicate the quality, like number of contigs, okay, or the size of the contigs, or and so on and so forth. So the contig number or count is simply the number of contigs produced in the assembly. This gives an idea of how fragmented the assembly might be. So fewer contigs would suggest a more contiguous assembly. Okay. Now, we have to be careful because an exceptionally low contig count might raise suspicion or doubts about some assembly errors. But generally, small number of contigs would mean something good. And you don't look at only one number, okay? We can look also at the assembly length, which is the sum of the length of all contigs. Okay, it provides an estimate of the genome size. And if the total assembly length is significantly shorter than the expected one based on our knowledge of the original genome, it might indicate lots of missing genomic region. We have N50, okay, the sequence length of shortest contig at 50% of the total genome. Here's an example to show you this, or the steps that achieve this, okay. This N50 is a statistic that provides information about the contiguity and size distribution of an assembly. It's kind of a weighted mean. So to compute this, one would first list all contigs or scaffolds from the shortest to the longest. From the shortest, you see these contigs, 30 base pairs, 40 base pairs, 50 base pairs. You arrange them from the shortest to the longest. Then starting from the longest, you would add up their lengths until we get into at least half of the bases in the assembly. So we know what is the full size of this. If you actually count the numbers, this is 170 plus 30 is 200, 60 plus 40, 300, 400. So we know that this full length of this is 400. This is the 400 here. So what we're going to do, we need to move from left to right now, okay, and then count, add them together until we get to 200. So 100 plus 70 is 170. We achieve the 50 here, or the 50% of the total genome, when we get to this contig, 60. So N50, that is the sequence length of the shortest contig at 50 of the total genome. In this case, for this example, N50 would equal 60. Okay, it is this sequence length in this case, okay? A higher N50 typically indicates a more contiguous assembly. But again, so it's really important to interpret this metric in the context, because again, an overly high N50 with a low contig count, if we have low contig and high N50, might suggest some issues, okay, like misassemblies and so on and so forth. I'm going to stop here and then we'll continue in the next part.